Hello everyone, and a warm welcome back to our Evolution and Ecology seminar series. My name is Elizabeth Duxbury, and I'm a postdoc in the research group of Alexei Maklikov at the University of East Anglia, and I'll be hosting today's session. As always, there will be a Q&A session directly after this talk, so please post your questions in the dedicated Slack channel and upvote the questions that you'd like to hear. And with that, it is my great pleasure to introduce today's speaker, Professor Dame Caroline Dean, Royal Society Professor at the John Innes Centre on the Norwich Research Park here in the UK. Professor Dean's research explores how plants sense temperature and epigenetically regulate the timing of their development, growth and flowering in response to cold exposure. During a remarkable 30-year career, Caroline's research has elegantly revealed novel mechanisms of temperature perception and epigenetic memory, including fascinating insights into how flowering plants are able to delay flowering until after a period of prolonged cold. Caroline's research has made substantial and important contributions to molecular evolution, epigenetics, and adaptation, in addition to the study of developmental timing in plants, and she has greatly expanded the breadth of genetic tools possible for the Arabidopsis thaliana plant model system. Caroline has received many prestigious awards throughout her career. To name just a few, these include an OBE, the 2016 Darwin Medal, the 2018 L'Oreal UNESCO Women in Science Award, and just this year, the Wolf Prize in Agriculture and the Royal Society's Royal Medal, testament to Caroline's outstanding research contributions. Today, Caroline will present her talk entitled Adapting to Different Winters, Noncoding Transcripts and Epigenetic Switches at FLC. So Caroline, thank you for accepting our invitation, and over to you.
Thank you, Elizabeth. It's a pleasure to be with you all. Yes, I'm going to talk about our work on how plants sense seasons. And the picture I show here of the trees is just a wonderful example of just how plants change what they do in different conditions. And if you think about the different conditions plants are exposed to in different parts of the world, that becomes even more remarkable. So at the beginning of my 30 years, yes, indeed, when I set up the lab in Norwich, I decided to tackle this big question of, just how do plants sense their environment and change what they do? And at that point, there was very little understanding. We knew that there were different developmental transitions that plants went through, and those were generally regulated by seasonal cues, the major ones being temperature and photoperiod. But we really didn't know how they worked at the molecular level, what were the major important cues for transitions from germination to vegetative development or from vegetative to the flowering stages. That was still very unknown. Now, we started off looking at the floral transition because it was an easy one to study. And also because I think it's really quite remarkable when you look at the bloom in spring, at just often how synchronized that is. And I show this picture of this row of cherry trees to illustrate that. All these trees have been through and exposed to these different winter conditions, and yet they've all undergone this transition to flowering at pretty much the same time. So how do they do that? That was the question that was always intriguing to me. And especially when you look at and think about the problem, winter, you think, oh, well, a prolonged period of cold. But the graph underneath the cherry trees here is a thermometer in a field in Sweden with a bird's eye view of what the temperature profile is, for an hourly profile over a year. And, when you think the plant is somehow integrating this noisy temperature to try and actually come from that a seasonal cue, or to understand that seasons are passing, to then make developmental decisions, then the process becomes even more fascinating. So we started off looking at flowering time because that was easy to follow. We used a molecular genetic approach and therefore used this reference plant, Arabidopsis thaliana. At the time, the molecular tools were not terribly well developed, and they're really much better now. But the questions we started off were, why do some Arabidopsis plants overwinter before they flower? And let me introduce that because, of course, most people think of Arabidopsis as rapid cycling. That's this type here on the right. where you basically, the plants just produce a small number of leaves and then flower, irrespective and not needing any winter period. But actually, the majority of Arabidopsis accessions collected around the world have what's called a winter annual habit, that they basically need prolonged cold to flower early. So the one question is, why do some have this winter annual habit and others not? This is known as a vernalization requirement. And vernalization requirements are a very important trait in many of our major crops to extend the geographical range. So the John Innes were very interested in that, because for winter wheat, for winter oilseed rape, of course, winter types are generally the ones sown in Northern Europe. And we had no molecular understanding of what the basis of that was. The second question was, for those types that do overwinter, how are the plants registering that they've been through the whole of winter and not get fooled by a kind of cold week in autumn, for example? And you seen here the plant under snow. The worst thing a plant can do is flower at the wrong time too early, and when winter has not ended, and then have snow. The reproductive success is then very poor. So those were two questions. And then the third question exploited the natural variation resource that Arabidopsis provided us. When you look in the stock centres, the Arabidopsis accessions now have been collected from very large parts of the world, shown in the red here on the map on the left. They've been collected from almost up to the Arctic Circle down to the equator. And it's an extraordinary resource that's still not terribly well exploited. When we looked at the vernalization requirement and the ability to respond to cold in these different accessions, you see this huge spread of natural variation, with some of them cycling very rapidly, these ones in the blue here, others being much later but responding to cold, and others being absolutely obligate requirers for winter and being very, very late flowering until they have received a period of cold as a young seedling. So the three questions then we were tackling: why do some need to overwinter? If they do, how do they overwinter? And what's this natural variation, and how has that allowed adaptation in these vernalization processes to adapt to these different climates? I had imagined would take us along a journey of temperature sensing, signal transduction, a whole series of things that you then find in textbooks responsible for environmental perception in all organisms, or even developmental timing, that you have some kind of signal, a sensor, whatever that may be. For temperature, we were imagining it might be a sensor in the membrane. There would then be a series signal transduction cascade, somehow to then influence genes in the genome to switch on or off to allow that developmental transition. That's what the expectation is. And in fact, people still address these complex traits expecting this kind of rather complex signal transduction pathway. Now, indeed, when people do analyses with mutant screens, with expression analysis, with genome association analysis for flowering time, many, many genes in Arabidopsis are involved. And I show a rather out-of-date schematic on the right here, just to make the point that many, many genes, when you mutate them, appear to, do influence flowering time. So then you put it into your flowering network. And there's been extensive analysis to try and integrate and understand this network. And that network looks, has different wiring, but similar components in these different plants. So this is what we were expecting to get into. In fact, we didn't get into a signal transduction pathway like that. All our genetics came down to was, in fact, we ended up focusing on one locus. So all of the questions about does this plant need to overwinter or not came down to understanding quantitative transcriptional regulation of this target. It's a gene called FLC, flowering locus C. It encodes a MADS-box transcriptional repressor, which basically stops the activation of all the genes in the meristome needed for that transition. So lots of genetics took us to transcriptional regulation of FLC for this overwintering requirement, question one. All of our analyses looking at look at the registration of winter, how plants actually perceive that winter period, took us now to a Polycom mediated epigenetic switch of this same target. So this gene is then switched off in an epigenetic manner by prolonged cold. And then a lot of our natural variation work to look at the adaptation of Arabidopsis and how vernalization and flowering has evolved to allow that adaptation, again, took us back to FLC. So not only did it take us back to FLC, this single target, but all our genetics kept taking us back to a chromatin mechanism, where I'm going to explain this chromatin mechanism briefly in my talk, where we have two states, an on and an off state. And actually, a lot of what we were looking at, whether there was an overwintering requirement, whether you could register, or whether you were naturally varying, influenced the switching between the on and the off state at this single gene. Now, I'm not going to get into very much chromatin, and if you look at the chromatin, this is a nucleosome with the DNA wrapped around. What's become very famous, and the cartoon on the left shows this, is these histone modifications that influence the histone tails, which then influence the accessibility and the regulation of the underlying DNA. You don't really need to know very much about it in this talk, except that some modifications turn the gene on, so we'll color code those green, and some turn it off, we'll color code them red. But what's interesting is it's a very simple little network combining transcription and in fact also non-coding transcripts that help switch the gene on and off. So what I'm going to do in my talk is give you a bit of an overview, not very detailed, about this epigenetic mechanism and how we've run into these non-coding RNAs. But then I want to get back to the topic that Elizabeth introduced, how are plants sensing the temperature and how is this integrated into this chromatin switching mechanism? I'll then briefly talk about how that mechanism has changed during adaptation, and then go back to, I think, what a question is I'd very much like to hear your thoughts on. Why, given we started from such three different questions, have we ended up focusing on one target gene? Because the expectation adaptation was going to be, it was lots of genes, lots of small changes combined together that was going to underlie these evolutionary changes that contributed to adaptation. So how come we've ended up predominantly looking at this one locus, how genetics has taken us to one gene, when there are clearly many that are involved and are required for the thing to flower? And then I'll just end up with some of the generic principles as we've studied FLC that now really we think about gene regulation differently because of what we've done. Okay, so we're going to talk a lot about FLC regulation. Remember, think of it as a brake to flowering. If the plant is expressing this protein, it stays vegetative. So it's a kind of double negative. You need to switch off a repressor to flower. So as the overwintering types germinate, FLC expression is very high in autumn. And this is just schematically shown at the top here. But as the plants begin to feel winter, FLC expression, shown here by looking at the RNA, is gradually switched off as the plants go through winter. And this is weeks and months of winter. FLC then stays off in spring. So this is this epigenetic aspect of it. All of the regulators, the positive regulators that are normally present in the warm are present in the plant here. But now, instead of being on, the gene stays off. So the question is, what's different up here in autumn when the plant is in the warm versus here when it's still in the warm, but it's experienced cold? So it's epigenetically silenced, and it stays silenced all the way through perceiving now warmer temperatures, along a photoperiod, all of the good things that help promote flowering. The plant flowers, shown schematically under here. This is an annual plant, but a version of this regulation is also important in perennials. But then what happens as the seed are produced on this plant, and this is what you're looking at in this box at the right here, these are the seed pods. And what we've marked here is the gene is marked with luciferase. So you can see this sort of funky pseudo color is expression of the protein, the FLC luciferase protein. And so you can see that in all of these little embryos, FLC is suddenly switched back on at a certain stage during seed development. And of course, it's switched back on because the plant wants then to start off from a high level again the following autumn. So FLC is all about talking about quantitative transcriptional regulation. That depends on, do you overwinter or do you rapid cycle? It's about shutting down the transcription early on in the cold, a switch now to this epigenetically silent state, which then is maintained through the spring and the summer. And then the gene is switched back on again to allow the whole cycle to go on every generation. So it's a beautiful example of an epigenetic switching mechanism, which actually takes a long time. So of course we can do time courses and all sorts of things, and the whole plant is involved. So there's, it's not like one cell in an embryo that's impossible to do biochemistry on. Okay, now I'm going to give a very curtailed view of a lot of what's going on, but the next, and I'm not going to have time to really get into this quantitative regulation, but the next 3 slides are all about our current understanding of transcriptional shutdown, the epigenetic switch, and the epigenetic memory. Okay, just a quick overview. Now, the transcriptional shutdown, absolutely, oh, I forgot to add, of course, I'm giving a real highlight here, but the last very detailed review we wrote was by Charlie Whitaker, a PhD student, and I would refer you to go back to this because we actually discuss about the evolution aspects quite a lot in this review. So I'd be interested to hear your thoughts, and if you want any more detail, I refer you to that. So this transcriptional shutdown took us into looking at these non-coding transcripts. Now, let me show this schematic here. On the top is FLC locus. It's about an 8 kb gene. These boxes here are the exon sequences. And what's at the top is what the sense RNA looks like. It's just a spliced version of all of the exons together. Not a lot of very interesting things. But at the poly A site of this sense transcript is a promoter for antisense transcript. So now it's the other strand that's made. And either the other strand is made without being spliced, or is spliced and polyadenylated early, what's called a proximal site, or is spliced and polyadenylated at a late site, actually upstream of the sense promoter. So these antisense transcripts completely envelop the sense transcriptional unit. Now, we got into this through actually some looking specifically for non-coding transcripts, but we're doing it in a very unbiased way. But the surprise was they go up enormously in abundance when plants experience cold. So this is some early work where we did a northern analysis. You can see a big increase in the amount. What we find when we look in each nucleus, and now in this panel in the middle here, you're looking at what's called a single molecule fish experiment in situ hybridization, where you take oligos and you coat single molecules of RNA. So you're looking at, in this nucleus here, green is showing you the antisense transcription. These are the probes. So here's a single molecule of the antisense transcript, and in red, you've got the sense probes. And what this showed us was that actually we get expression in the cold. I haven't given you this data, but actually that expression is never at the same locus. So if you look at the sense and the antisense, what's happening is that each allele makes either one strand or the other. And that because the antisense goes up in the cold, that shuts off the other strand transcription. So the very first stages of the cold are a transcriptional shutdown because the other strand is made. So that's the first step. The second step is this epigenetic switch. And again, very different dynamics, takes much longer. It's got other cold factors involved, one of which is a gene called VIN3. VIN3 encodes a protein with this kind of domain structure, doesn't really matter. It's an interesting PLC2 accessory protein. It comes along with a Polycom complex called Polycom repressive complex 2. This is a very conserved chromatin complex that's found in all high eukaryotes and that is involved in conferring memory. It was discovered in flies, Drosophila. And so what happens in flies is that as the flies are developing, transcriptional regulators switch a gene on, but those regulators often go away. So the Polycom then comes along and fixes that expression state. And it's involved in K20. Its main biochemistry is that it methylates lysine 27 with a trimethylated group. So you have this sort of alphabetic spaghetti in chromatin world. Histone 3, K for lysine, the position 27 is trimethylated on the tail. And that's this red blob. Okay, so this means the gene is off. And this is epigenetic. So the Polycom complex that delivers K27 trimethylation is recruited to it to allow this thing to be stably inherited. So what we find is that this cold-induced protein, Vin3, partners with something which is a homolog of itself, but it's not cold, it's constitutively expressed, called Vern5. And together they bring the Polycom repressive complex 2 to FLC at one site here in the middle of the gene. It's not actually in the promoter. And you get this then, the actual switching process involves nucleation of this Polycom complex. That maintains silencing, but actually for long-term silencing now, when the plants come into the warm again, this Polycom complex has to spread all the way across the gene. So in the cold, it's just at one site. When you come into the warm, because now the plant's dividing very rapidly, to maintain epigenetic memory, you have to have this spread state. Now, I'm at this point not going to go into very much more mechanism, just one more thing, and then we'll get back to evolution. But I'm going to talk about how this mechanism has changed. So I just needed to explain the core mechanism to you. So I've talked a lot about this Polycom complex. And in fact, if you look at what that Polycom complex delivers, this H3K27 trimethylation at the locus. The locus is along the bottom here. We track all the way along. This nucleation region is this red bar here, just downstream of the promoter. And you can see that during the cold, when we look at the accumulation of K27 trimethylation with increasing weeks of cold, you see this progressive accumulation. Now that fitted with the very slow progressive silencing that I showed you that happened during the cold. That's actually during the cold, when the Polycom is just at that internal site. When the plants then come back into the warm, Polycom spreads now across the whole body of the locus. And again, you see it's quantitative. The more cold, the more K27 trimethylation at the locus. And so the first interpretation of this was, well, quantitative chromatin modification is what explains the quantitative silencing that we see in the cold. But actually that's completely wrong. It turns out that's a problem with a population average because, of course, we grind up lots of cells to get this kind of assay to work. What is actually happening is that there's a switching mechanism. So that now what I'm drawing you is this green and red for the different states of chromatin at FLC. And I've drawn it schematically on roots because I'm going to show you some root data. Now, people say to me, okay, Caroline, now you've gone mad. We're talking about flowering, and now you're showing me roots. But actually, vernalization is a whole plant phenotype or trait, and that the roots are really very important for perceiving the temperature, which then transmit through actually a mobile protein called FT, which is a downstream target of FLC, so whatever's happening in the bits around that is feeling the temperature is then integrated through the whole plant by movement of this FT protein. So as plants initially growing in autumn with no weeks of cold, all of the cells FLC is on, so I've color-coded them green. But as the plants then experience more and more cold, during the cold, you have this switch, which is the nucleation of this Polycom complex at this one site in the gene. And that switch is happening in each cell. In fact, it's happening at each allele independently, but I haven't drawn that here. But as the plants are now experiencing more and more weeks of cold, more and more cells have switched. And it's a relatively low probability switch. which is why vernalization takes weeks and doesn't happen in a couple of hours. Now, this is in the cold. When you then bring the plants, and there's not that much cell division in the cold, when you bring the plants back out into the warm, now the roots grow. And in fact, why the roots are so good to image is that the meristem, which is down here, the cells in the meristem divide in this anticlinal mode so that this column of cells is a developmental lineage from the meristem cell down here. So whatever state has arisen in the meristem in the cold, as the plants then grow out in the spring and the summer, all the daughters and granddaughters of that cell remain in the on or the off state. And so as more and more cells have switched, as they grow out, more and more of those cells then remain in the off state. And of course, when you grind these up, you have a beautiful graded change, but it's not graded at the cellular or in fact the allele level. Now, let me show you, actually, this is the schematic. When you then look at the plants, this is what you see in real time. Here's an FLC venous fusion where we're looking now with no cold. All the cells are expressing venous, but with increasing amounts of cold, you see more and more of the files have switched FLC off, and by the end of 10 weeks, they're all switched off. So what we've got here is, if I summarize what's going on, is in the first phase of cold, we have the sensing, we have the antisense pulse, which transcriptionally switches the gene off. A bit slower, and with accumulation based on the Vin3 dynamics, that's one of these accessory proteins in this PRC2 complex, you nucleate, which is a digital nucleation, which is then switching that allele to remain off, and that then spreads across the body to help maintain that as a long-term memory during the spring and the summer. And then it's switched back on again. In fact, with the reversal of this, it's a demethylase of this same modification. Okay, so that's the mechanism. And what I'm going to do now is focus a little bit more on this temperature side, because there's various temperature aspects I've told you about so far, the upregulation of the antisense, but also this slower upregulation of this PhD accessory protein called Vin3. And it was always quite interesting, you know, how this very complex temperature profile I showed of winter was being integrated to actually affect the regulation of these different factors. And so about 5 years ago, funded fantastically by the ERC, it's one of those blue skies things. They let me do a field experiment. You know, not many agencies would have done that. So hats off to them. We set up field experiments with collaborators, and I'll show you their pictures. Svante Holm from Mid-Sweden University, who was the engine of this whole experiment really in Sweden, and Torbjörn Sahl, who's at Lund University. And they were absolutely instrumental. And so they had two master students, Christina and Katja, who did the work in Sweden. And then we cannibalized a greenhouse in Norwich to sort of pretend it was a Norwich field with no heating and no lighting, but at least we could add some transgenic plants in there as well. So we've got some mutants and transgenic lines, which of course we couldn't put in the field. And, you know, poor old Christina, as you'll see here, she had to dig, you know, often 3 feet of snow. So we planted everything in September time, and then almost weekly, the students would go out, harvest leaf material, and then send it back to us so we could do the expression analysis over the whole of winter. And we did this over 2 different winters, and we were very lucky. We picked 2 very different winters. Then that's the data I'm going to show you. So what did we see? Well, I mean, one thing that was happy to start with was, yes, in fact, we got very different winters. Here's the temperature profile in Norwich. We had 2 plantings in Sweden, which actually usually the temperature profile is rather similar, and then South Sweden. And so we ended up with, in this particular year, it was different. In some years, we managed to get really very warm. We had one very warm Scandinavian winter we managed to capture. In Norwich in this year, this was 2014, there was a very warm autumn. And it took a long time before the temperatures dropped. But what we did see, and this is now looking at either Vin3 accumulation, which is the boxes over days from sowing, or actually FLC reduction, and the data is plotted here, and then the sort of the mass is looking at calculating the rate shown in the line here. So happily, FLC went down and Vin3 went up, which, you know, is always reassuring. That's what happens in our cabinets and our greenhouse, but, you know, actually in the field, it could have been entirely different, but no, it wasn't. And so what I'm going to do is focus in a little bit on the Norwich data. So this is the same box as you saw before. And in fact, as we looked at the Norwich data, we realized that in fact, the FLC decay almost looked like there were two rates, an early rate and a later rate that seemed to tie in with the when Vin3 was expressed. And that fitted when we looked at the Vin3 mutant, because the Vin3 mutant, the FLC decay rate didn't switch to this faster rate at a certain point. So this then defined, and it was this warm autumn in Norwich that helped define this, that actually there were different rates depending on Vin3 accumulation. And Vin3 is this temperature dependent one, if you remember. So then we ended up saying, okay, well, how is this fluctuating temperature profile upregulating these factors? So we set up some chambers back here in Norwich where we could recreate the field data, but in a chamber, the field temperatures. So this is what the plants saw every day. They either had two temperature regimes of a mean around 8 or fluctuating 8 with a mean over 24 hours of 8, or separately, and that reflected the Swedish winter. The Norwich winter was a mean of 15, but actually we also gave it a fluctuating around a mean of 15, which went up to 22 and down to 8, I think it was. And then we gave the plants this fluctuation for periods of time and then looked at what was going on with Vin3. Bit complicated, I know, but this was such a crucial experiment for us. This came out in 2018. Because what it told us was that in either a fluctuating or a constant condition around a mean temperature of 8, there was exactly the same Vin3 accumulation over time. But actually, when we looked around a fluctuating 14, there was a difference in Vin3 accumulation. And in fact, with this fluctuating temperature, we got very little accumulation of Vin3. Now, historically, people had always assumed that plants were going to be time averaging and integrating temperature over periods, you know, sort of reasonable periods of time. But this was over a 24-hour period, this fluctuation was affecting Vin3 upregulation. And in contrast, it was exactly the opposite for the shutdown of FLC itself. And I cut straight to the chase here. Fluctuating 14, actually here, the fluctuating temperatures promoted the shutdown. So for Vin3, you needed absence of the warmth in the afternoon. For FLC, we needed the presence of the cold. So when I plot that out onto what the plants are seeing, what that told us was that the nighttime temperatures were triggering these antisense dynamics, which was shutting off the transcription I was telling you about, this early phase of the cold. But these much longer term induction of Vin3 required temperatures that were under 15, and afternoon temperatures of anything over 15 was turning over all the Vin3 that was being had been synthesized. So in fact, the plants are not only looking at cold temperature exposure, they're making sure that the warmth has gone away as well. So it's absence of spikes in the afternoon in autumn is as important to know that winter's going by as presence of cold, you know, freezing temperatures at night. And that was a completely new aspect to our thinking. Now we've gone on a lot more, and this has come out in a cell systems paper by Rea Antonio Cuinato. It's a theory paper, but it basically looks at temperature sensing in this whole system. And what we find is that contrary to what was in the field, that there would be major thermosensors. You remember my cartoon at the beginning that there'd be a sensor in the membrane triggering a signal transduction pathway. That was the sort of paradigm that's out there. But actually temperature affects every aspect we look at in this system. And different aspects of the temperature affect different components. So you can't really think of a major thermosensor. All the steps are thermosensitive and respond to different aspects of the temperature. And it's the integration of all of that through, first of all, Vin3 expression, and then how VIN3 works on FLC that gives you the overall temperature profile. But Rhea has now, and what this paper shows is actually a model which now integrates all of this that can predict what FLC will do in a new field experiment. So she can then project out, climate change scenarios, and actually with this mechanistic understanding, actually predict what it's going to do to vernalization. So I recommend you read that one. Now, a very recent paper that's just come out, we went into all of the mechanism behind this long-term cold sensing. And I have to say, I'm sorry, I don't have any time to get into this, but this turns out to be growth. The long-term sensor is how rapidly the plants are growing. And it all comes down to a very stable protein, which upregulates VIN3, which accumulates slowly in the cold when cells don't divide. But as soon as they divide, it dilutes. So in fact, what affects that particular factor is growth rate. And so that is the long-term sensor. Anyway, if you're interested in that, this is the reference to that paper. Because I want to get back to more the evolutionary aspect, because we can now look at the mechanism and say which facets have changed upon adaptation that I showed you in the natural variants of Arabidopsis. A long, long time ago, we collected all those natural accessions and looked at their response to different periods of cold. And I just show a few here. What was striking was that some of them, and characterized by one here from Edinburgh, if you gave the plants 4 weeks of cold, it pretty much saturated the vernalization response. So this is a measure of flowering leaf number. This is the acceleration with increasing cold exposure. You can see it's flowering with much lower leaf number, i.e. much earlier after 4 weeks, and then plateaus, no more effect. Whereas some of these other ones, particularly the Swedish ones, this one here, this orange one, you gave it 4 weeks cold and it barely noticed. It needed very much longer cold to vernalize. This was from northern Sweden, and I'll show you what that looks like. So what was clear was that the length of cold was a variable that seemed to fit with adaptation, so that the accessions that needed longer cold were from all of the northern latitudes. Just catch myself up here. Now, of course, we've got a lot of these accessions. We've got the phenotypic data. We've also got a lot of sequence data now, but in a step before that, we had SNP data, so we could do sort of a population analysis of what was going on. And if we looked around FLC at, this was in the 1300 Arabidopsis accessions, worldwide accessions, at different genotypes, so different, each line here, and there are 1300 in a row, shows you the SNP chip results of that accession. And you want to home into the 50 kb around FLC, which is between these two black lines. And what you can see is that there are groups color-coded here, and there aren't that many haplotypes actually in the worldwide set, sort of a handful, some major ones, about 6 or 7 major haplotypes in the worldwide population, but that these extended quite a long way along the genome, suggesting that they were selected and maintained so that there was not so much recombination, because this is quite a long haplotype for Arabidopsis. But what you'll see is we characterize them based on whether they were a slow vernalizer, SV, or a rapid vernalizer. And this is an SV, this is an SV, an SV, and an RV. What you can see is that actually slow vernalization was attributed to quite different haplotypes. So in fact, early on, we were quite disappointed because everybody tends to do genome-wide association as a way of identifying the factors involved in their traits. GWAS does not pick up FLC as a major important gene underlying adaptation of vernalization, because you have this allelic heterogeneity reduces your GWAS signal. So I just put this in as a cautionary note that all our QTL analysis pointed us to FLC, underpinning all of this variation. I forgot to say that from the previous slide, but GWAS analysis didn't. But so these haplotypes, there's no obvious geographical distribution to sort of have a classic cline, but it's much more complicated than that. And as we went into it, and we looked in more detail now, and these are a subset of those groups, you can see that there are groups which are very similar to each other and quite separate from other groups. But all of that variation turns out to be non-coding. All of these genes encode exactly the same protein, FLC protein. Now, I've put these colored circles around to make it simpler to understand what I'm going to say. That's when we then looked at FLC expression in those accessions, we then took that gene out and put it into a common background. So we took away all of the sort of complexity of the other genes that might be affecting FLC. So this is now on the bottom here, the expression of those allele types, haplotypes in a common background in transgenics. What you can see is that correlation is very good. That's saying that most of the non-coding variation is what's defining these different behaviors of FLC. So what we've got then in this worldwide set is a few major haplotypes defined by non-coding SNPs. And so the assumption was they were conferring some advantage. So we then go back into our field experiments and say, well, what advantage are they conferring? Now, again, you know, there's a massive data here, and I give you a little snapshot, but this has just come out in eLife. So please, I refer you to this paper down here. But if you remember that, as I was describing before, we have FLC starting levels, which I haven't shown here, but then there are these two rates of silencing dependent on this VIN3 timing of the induction. So we could then go in and score those three aspects, starting levels of FLC, the rate of silencing in the early VIN3 independent phase, and the rate of silencing in the VIN3 dependent phase in the accession, or actually a near isogenic line where we've tried to simplify, so we've only got FLC as the variable. To cut a long story short, and as I say, please refer to that paper, the most significant aspect that is varying, that is important for the field adaptation, is the starting level of FLC and the very early phase of silencing. And it's the nils have the same behavior as the accession. So it's these non-coding SNPs at FLC that are defining that phenotype, that phenotypic difference. I just show you some here. For example, here's FLC expression days in the field. Here's a rapid vernalizer, one in black. In blue and in dashed lines are this Lervik accession, which is this northern Swedish one, and the near isogenic line where we've introgressed just FLC into this this Cull-Fry background. So there's the only difference is the FLC locus, and you see then it's very much higher expression early, and it takes longer to come down. And what we find is that actually the biggest difference in terms of they've all actually vernalized very efficiently by December. So the FLC silencing is probably not the variable that's driving the behavior in the field in terms of maintaining these different haplotypes. But what we do find is that what you absolutely have to have is this high FLC expression in places where you could have precocious flowering, and that was in Norwich in 2014, and these early, in fact, in Sweden in 2016, we had very warm autumn. It was the year that the seed bank flooded. If you remember, there was this very warm autumn. There were actually plants in our field trial that flowered. And of course, then they hit a northern Scandinavian Sweden winter afterwards. And it was really atrocious for fitness. So basically, the selective advantage is that these alleles are keeping FLC high and keeping the early silencing very inefficient to make sure that the plant has got through the whole of autumn and has had the full winter. Okay, now I'm just going to speed up a little bit. This just shows you the population I'm going to talk about. One of them is this Swedish population. Actually, I'm probably slightly running out of time, but let me just show you a few pictures. This is a very stable population on a beach in southern Sweden. And this is Svante Holm and Torbjörn Sala. And we had some wonderful field trips doing all of this, but there's a wonderful population there that is crying out for ecology. And now we've got all the molecular details behind it. I'm going to miss these out because but this is the northern Swedish field site right at the high coast in northern Sweden. This is a World Heritage Site. And again, a very stable population. You know, people think of Arabidopsis as this sort of model plant, but these are stable populations that have been studied for many, many years by Svante Holm, and they're wonderful to do the ecology and the evolution on. I will just go into this Lervik one. So Lervik, I've told you, is this one that you give 4 weeks of cold and it barely notices because it's adapted to this northern Scandinavian winter. In the experiments in the lab, you can show that FLC goes down with the 4 weeks in the cold. And in our normal rapid vernalizing types, 4 weeks is enough to keep FLC off. But actually, the FLC from Lervik will come back on. And in fact, what you need to do is give this plant 12 weeks of cold to get the full epigenetic silencing. And when we actually go in and define what's going on, and these schematics, I hope you remember from the mechanistic part, normally, you know, you have transcription shutdown, nucleation, spreading. In fact, this is what happens in the rapid vernalizing type. In this Lervik type, you have exactly the same for during the 4 weeks of the cold, for certainly a certain period as the plants move in the warm for 10 days. But once the plants have been in the warm a bit longer, the silencing is lost. And you need 8 weeks of cold for the silencing to be maintained for 30 days post-cold. And we've now gone in, and this is this last paper here that you'll see, this Quester et al. We've defined a single SNP that defines this stability of this long-term silencing. It's all to do with the battle between transcription trying to switch the gene back on and the epigenetic silencing maintaining it in this post-cold stage. But, you know, this is remarkable. This is a single gene, single non-coding SNP driving the behavior of this particular FLC type, which is so important for allowing this Arabidopsis to adapt to northern Sweden. Okay, so let me just tie up. I've told you there are these different haplotypes. We've got good information on 2. We've got a whole lot more to do, so it's not anywhere near the end yet. But I think these natural SNPs, which have evolved in the haplotypes, are wonderful ways of dissecting the mechanism, because evolution's done the mutagenesis for us, if you like, in terms of which SNPs are going to be driving molecular variation, which gives us a phenotype. And then I want to go back to this question, which hopefully we'll have a bit of discussion about. You know, here we were talking about flowering and vernalization and temperature response. This is a hugely complicated set of processes in a plant, but all our genetics kept taking us back to this single gene target. And why is that? And I argue that, in fact, this gene is regulated by trans factors, turning the gene on and off, switching it on and off epigenetically, that are regulating thousands of targets in the genome. I haven't really gone into what those targets are, but nothing we got out of any of our screens was flowering specific. So this is a gene regulated by trans factors regulating a large proportion of the genome. So in fact, to actually get variation, what you need is cis variation in response to those trans factors. So then you drive the cis variation, changing maybe binding affinity of a trans factor or something, which will then give you molecular variation, which is then also interestingly translated into phenotypic variation. So that's also an important component in here. And because FLC, any molecular variation, is driven into phenotypic variation, that's what has allowed this wide adaptation with these very different FLC starting levels. This is a color coding here of the starting level of FLC from the accessions collected from all over Europe. So I would argue that, in fact, this antisense circuitry has given this huge target for cis variation to have arisen fairly frequently. And because the cis variation feeds into a chromatin mechanism, which feeds back on itself, you can vary lots of things to have the same outcome. You can affect transcription in different ways. You can affect the epigenetic silencing in different ways. So this has been a large template, and this is why FLC has become the node. And actually, we've got one recent paper on the vegetable brassicas. It's the same story in the vegetable brassicas. They just have multiple copies of FLC. So let me wrap up. You know, what have I tried to explain to you? Well, our starting point was to follow up this wonderful question of how are plants sensing their environment, specifically the seasons. And we focused on flowering and focused on this requirement for winter as a way of being able to dissect that. And the surprise has been that it's taken us into this core antisense chromatin circuitry. And I argue that it's because of that circuitry that this gene has become a node for variation for flowering time and vernalization. And that the adaptation is mainly through these non-coding SNPs. That the haplotypes are selected out there because they are maintaining high FLC expression, because what absolutely is selected for is you don't want to prematurely flower in autumn. So you've got to stop premature FLC silencing. And that the temperature aspects, instead of one or two major temperature thermosensors in the system, is that actually it's this very noisy temperature profile is integrated through many different aspects, through peaks, through troughs, as well as long-term sensing through mechanisms as unexpected as growth itself. But we've also ended up discovering an awful lot about gene regulation. As I said, none of these things that regulate FLC are specific to flowering. So what we've ended up becoming very focused on is how chromatin and trans factors talk to non-coding transcripts to set up chromatin environment states, which then determine what the gene does in terms of amount of expression and the ability to switch. Now, this might be a bit too much, but I just added at the end for those who are interested in the mechanism. Most people in a textbook think genes are regulated via a classic trans factor switching on, finding the cis element in the promoter and switching the gene on. I would argue, and this is what's all in the textbooks, that this is probably wrong. It's conferring qualitative expression regulation, that mechanism, but really it's the local chromatin states that are driving quantitative variation in expression. And I think quantitative variation is going to be, transcriptional level is going to be very important in many aspects of evolution and ecology. So, local chromatin states are important, and the chromatin environment, but it's in a very complicated feedback loop. There's never an upstream and a downstream. Everything feeds back on itself. That actually everything is a tug of war, that you don't have a sort of nice positive switching on. There's always a positive and a negative, and gene regulation is all about that tug of war and changing one side or the other. And that there's non-coding transcription, which is hard to see. I sort of propose it as the iceberg. The RNAs that we see are the tip above the water, and you have this huge bit underneath the water, which is turned over so rapidly that in most RNA-seq or expression analysis, you just don't see them. But those are what are driving the chromatin regulation, which is driving the transcription state. So, look out for non-coding transcription. I think it's going to be very important for gene regulation generally as we go forward. So let me just acknowledge the group. I've picked a relatively old picture here, mainly because this is in southern Sweden at a wonderful place called Drakamölen next to our southern Swedish site. And this is the host, Ingelil Thorsel. She was a wonderful host. And these are the people who were involved in the field experiment. We used to use it as our lab retreat. Here's Svante and Torbjörn, who would join us, Rhea, who's done a lot of the modeling, and Joe Hepworth did a lot of the field work. And Charlie is the one who wrote the review I talked about. And thank you to our funders, and thank you to you. And I hope I haven't used all my time up. I hope we have time for questions. Thank you.
Thank you very much, Caroline. That was a really fascinating talk. And yeah, definitely some very fascinating insights into gene regulation and novel mechanisms via epigenetics, as you describe in this non-coding regulation. So we started to have some questions coming in from our audience. And I also thought I would kick off with one of my own. So I was quite curious about the effects of climate change on these mechanisms. You talked a bit about how you'd employed a modeling approach as well to address this question, and how with the effect of warming winters perhaps, or changes in periods of cold spell, and how that might have a knock-on effect on flowering. I wonder if you could talk more a bit about that.
Well, I would encourage you to invite Rhea to give you one of your seminars, because this is exactly what she's now setting up her own independent lab to try and do, to sort of use a modeling approach to build on the mechanism, because what's happened before is the modeling has only been a bit abstract and we haven't really had the kind of mechanism to go on. So with her thermosensing through the system, she's now trying to integrate the experiments with exactly what would happen. And so she's done a 50 years from now projection, and it's not all bad. In fact, in the UK, actually vernalization is not terribly affected, but of course in different parts of the world you have very different effects, and she can map all of that out for you. So there isn't a sort of one simple bottom line. It's not all bad and not all good, but depending on where the plant is growing, will have a very large effect of climate change.
Great, that's really interesting. I guess it reveals the whole complexity of the system.
Yeah, it's different in different places, and because of the complexity of the system, and it's different, and different parts of the temperature different parts of the temperature profile are picked up by different parts of the system.
Yeah, exactly. That's really interesting. So we have a question which asks, since one copy of FLC turns off the other copy of FLC, and there are different alleles of FLC, do you know what happens in heterozygotes, and is it always the same allele that turns off the other, and would this be related to...
Let me just correct. One allele doesn't switch off the other. The antisense of one allele switches off the sense of its own allele.
Yeah.
So it's the antisense switching off the sense. The two alleles behave independently. And in genetics and in molecular experiments, you can see they switch independently. And if you look at some of those early papers, we show all of that quite clearly with the Venus experiments and so on.
That's interesting. Thank you. So there's a question which asks, what, if anything, prevents adaptation through non-synonymous SNPs, and would the changes be too strong?
Yeah. It's always very hard. I mean, presumably, the amino, there's been selection to maintain that amino acid composition, so that the non-synonymous SNPs, you have been selected against, presumably, because there are many examples where, have I got the question right? The non-synonymous, where it would change the amino acid, you mean? Yeah. So presumably FLC amino acid variation is selected against. But there are many examples. So Frigida, which I didn't get into, which upregulates FLC, there's a lot of synonymous. Which way around is it? It's non-synonymous changes the amino acid, right? Which way around is it? I forget. It's not something I do think of very often. But anyway, amino acid variation often occurs in many different proteins. It's just not apparent in FLC for some reason, presumably because it's selected against.
I see. That's really interesting. And it's fascinating how it is this single locus, the FLC, which is like the target as you describe, where the epigenetic silencing is focused on. I think that's really fascinating. I wonder whether other processes related to regulation could also have this very genetic base.
I hear talks occasionally by David Kingsley, who works on fish evolution. Now, I'm way out of my field here, but fishes lost their hind limbs, apparently recurrently, the evolution of fish. And he has a very similar story. It's the same loss of function of something, which has given this independent evolution by loss of limb. So, in this sort of this whole idea that evolution only occurs by small changes in lots of targets, I think actually needs to be revisited a bit with the new data sets that are coming out.
Definitely. And I think that's definitely a question to address in lots of different contexts to see.
I would love someone to turn their thing on and tell me some examples. I mean, I'm looking, I pitched for the evolutionary side of my talk to hopefully get some feedback.
Interesting. So yeah, we have a couple more questions actually, which have just come in. So one asks, you emphasize how plants don't want to flower too soon. Does flowering too early affect survival of the plant or only?
Yes, absolutely. I think I showed a picture of the plant on the snow. You know, the flowers don't finish forming, the seed don't form. Reproductive success is a big fat zero. So plants have got to make sure that they flower and set seed in favorable conditions, which is why we have the spring bloom, you know. That maximizes reproductive success.
Yeah, exactly. So it's really important for fitness for this to be tightly regulated. And there's one more question which asks, I'm wondering whether FLC targets, I'm wondering what FLC targets in the downstream physiology. Yes. And about the associated cis SNP. Can we really find one SNP or several haplotypes control the expression of FLC? And can we design experiments on haplotypes?
Well, as I showed you, there are 10 or so haplotypes. There was a Lee et al. 2014 Genes in Development paper which define all the haplotypes. Those are characterized by non-coding SNPs, but you know, between 25 and 40, depending on which comparison you do. But yes, indeed, we've gone in, for one I whizzed through too quickly, so I was running out of time, but the one I showed you, we've defined by mix and matching and introducing one SNP back in to show that it recreates the phenotype. So I apologize, I couldn't really get into the details, but please do go and read the papers and you'll see the data.
Great, definitely. Lots to explore further for sure. I'm afraid actually that's all we have time for today. But thank you very much, Caroline, once again for a very fascinating talk. Thank you.
My pleasure. Okay. Thank you, everybody. Please email me. I'm very curious, you know, wacky thoughts sort of from I'm not an evolutionary biologist and I'm not an ecologist, so I'd be very interested in any feedback.
Perfect. Thank you. Great. So just before we finish, I'd like to remind everyone that next week our speaker will be Professor Ruth Mace, and she will be talking about the behavioural ecology of religious belief and practice. So something a little bit different. Thanks very much again for tuning in, and until next time, stay safe and see you again soon.